Okay, so actually all this stuff's published. Anyway, so life on Earth has been hacked, I'm going to tell you, and for all time. And life is made of little genetic Lego RNA of known sequence. That was a surprise to me. tRNA evolution theorem was not deeply encrypted. So if God was keeping secrets, they were not that hard to hack, as I hope to convey. So how does pre-life selection work before Darwinian selection, before you have real organisms? Try and address that to an extent. And what's the driving force? And I'm going to say that's polyglycine, that life evolved initially to synthesize polyglycine. That's a driving force behind pre-life selection. And the approach that I took was top-down, so sequence-based approach. Bottom-up approach would be making life in a test tube, and I think at this point you can at least consider doing middle-out type approaches to origin of life. So evolution of tRNA provides the core explanation of the origin of life. If you evolve tRNA, you get life, and if you fail, then you fail to get life. The evolutionary analysis can be used to mend the alignments of the tRNA D loop. These are not too bad the way they're currently devised, but they could be improved, and also the D loop. These are not good and they need revision. Better alignments will allow full analysis of tRNAs through the tRNA sphere, so all the tRNAs in the database at least. And with AI to insert missing data, you can extend tRNA modification data, and that would be another potential advance. So I think what I'm telling you is enabling. What I want to convey to you is that tRNA evolution is solved to the last nucleotide. tRNA evolved from RNA repeats, so GCG, CGC, 5' acceptor stem, 3' acceptor stem, UAGCC, that's the D loop, stem, loop, stem. And this is a specialized loop, a 7 nucleotide loop with a U-turn, a slash after a U. The C interacts with the A and then stacks on the stem. Wobble, Watson Crick, Watson Crick. And this one is part of the reading of that one. So we'll deal with that in some detail. And tRNA was generated from ligation of 331 nucleotide mini helices of almost completely known sequence. The only exception is the 3 that were scrambled in coding. Okay, the rest of the nucleotides are known. The pre-life world was capable of complementary replication, RNA ligation, ribozyme nucleases, internal RNA processing, and generating RNA repeats. So it's a complicated world that we're trying to describe, based mostly on tRNA sequence, actually. tRNA is a living fossil from 4.2 billion years ago. The exact dates matter, but I don't know them. That's an approximation. And tRNA evolution is the core story in evolution of life on our planet. So many thanks to Jerry for inviting me and the genetics program for hosting me. It's really lovely to be here. This is the tRNA molecule, and I'm not sure it's well described in textbooks and in the literature to an extent. tRNA is a molecular fossil from 4.2 billion years ago. And what I'm going to show you is about a 4.2 billion year old puzzle that's solved for all time. So it was a surprise to me that this puzzle was as easy as it was to solve. And I'll try to convey that in the seminar. tRNA is the genetic adapter. It's where amino acid sequence meets nucleic acid sequence. So here you have wobble, Watson Crick, and in mRNA you have Watson-Crick, Watson-Crick wobble. Think of coding in terms of tRNA first, mRNA second, storage in DNA third. Let's see. It's a natural biopolymer with surprising properties and an inscribed history. So the evolutionary history of this molecule is written into its sequence in a way that it can be revealed. It's really difficult to redesign this molecule. So if you want to improve on the genetic adapter on Earth in order to create a master race, let's say, that's actually a difficult problem. It's hard to redesign, for instance, this loop, which I'll describe in some detail. Life evolved around tRNA and the tRNA anticodon. I'm going to say that ACCA gly is the primitive adapter. So ACCA with glycine on the 3' O here. That can be combined actually with an extensive extension of the screen segment. And you can run wet-dry cycles, and I bet you can make polyglycine with a glycine here. And I bet that would work in your laboratory using published protocols. Okay, so ACCA gly is the primitive adapter. It gets attached to various RNAs, including tRNA, as I'll describe. And the elbow, that's where the D loop interacts with the T loop. So describe this in some detail. This is the Levitt reverse Watson-Crick base pair. A reverse Watson-Crick base pair is like Watson-Crick GC pair in this case in DNA with one of the bases flipped over. So it forms 2 hydrogen bonds, not 3. This should have a fancy name, but it doesn't. The original sequence is 100% patterned. So here you have a stem loop stem, 17 nucleotides, and a homologous stem loop stem here. The sequence and that sequence were either identical in pre-life or complementary. It's hard to tell because the stem loop stem sequence reads almost identically forward and back. That's a GCG repeat, a CGC repeat, a UAGCC repeat to form the D loop. The original sequence is 100% pattern, and so you're enjoined thus. Okay, so chemical evolution of life on Earth. What I'm going to say is that if you evolve type 1 tRNA and type 2 tRNA with this end loop here, you get life. And if you fail in any of those attempts, you will fail. This molecule, type 1 tRNA, relates very closely to this type 2 tRNA. Here you have the V arm, this chicken wing. That's 7 yellow linked to 7 green. Here it's 5 yellow linked to 5 cyan. This is linked to 5 cyan. So if you delete 2 yellow and 7 green from an early version of this molecule, you create that molecule there. This is made out of ancient RNA Lego. That's ancient RNA Lego. And this loop was evolved in pre-life, selected in pre-life, as I'll try to describe. Okay, life evolved around tRNA and the tRNA anticodon. And life evolved chemically from little genetic Lego RNA. And this was a surprise, at least to me. I wasn't expecting this. And I think this is the reason this was the fastest way to do it. If you try to evolve this molecule and this molecule by some kind of a chaotic process, you'll fail. tRNA was the birth of biology. tRNA evolution is solved. And tRNA evolution has consequences for evolution of life on Earth and evolution of the genetic code. So this is published, but I'll just touch on it in this talk because of time constraint. So this is an attempt to go between this little Lego construct over here and sequence. And this used to be really easy to do, and it's harder now, and that's a little bit sad. But this is Pyrococcus furiosus. You can think of this as LUCA, the last universal common cellular ancestor. And it's 46 sequences. That's a complete tRNAome in an ancient organism. And it's a typical tRNA sequence, which is like a consensus sequence. So I produced this output with about 3 clicks of a mouse and was sort of stunned to realize, I didn't have the arrows or the arms here, but I was sort of stunned to realize that this stem loop stem, the T stem loop stem, the anticodon stem loop stem were homologs. I think if you stare at them for a moment, you'll concur. So the typical sequence is C, C, G, 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 C, C, G, G, that can be G, 7-mer loop, U-turn after a U, 7-mer loop, U-turn after a loop, a U. So this stem loop stem and this stem loop stem, 17 nucleotides, stems of 5, 5 plus 5 is 10, plus 7 is 17. What I'm telling you is that this, after 4.2 billion years, is an obvious homolog of that. And Pyrococcus furiosus is a record of LUCA, the last universal common ancestor. On the Lego image here, stem loop stem, homologous stem loop stem, as it's colored. That's 17, and it turns out this interval here is 17. And this interval here, that's the D loop, is based on a UAGCC repeat. So UAGCC, UAGCC, U, G for A, GCC, UA. So this is like the only break from the pattern here. And that is a G and not an A because it intercalates between this A and that A and hydrogen bonds to this U just before the U-turn. And that's that base there. That's the intercalating base. It's a G. It was an A in the initial pattern. It's between, in this case, a G, often an A, and another A, hydrogen bonding to a U just before the U-turn. Right next to it is a G off the rack that interacts with a C. That's the G, and that's a C. That's a Watson-Crick pair, 3 hydrogen bonds right there. These 5 here, let's see, I should do these first. So this is the 5' acceptor stem, 3' acceptor stem, 5' acceptor stem, 3' acceptor stem. That's based on a GCG repeat and a CGC repeat. So after 4.2 billion years, GCG, GCG, G. And it's CGC complement. These 5 here are those 5 there in green. They correspond to these 5 in green. And that should have the sequence G, G, C, G, G. It's got the sequence G, that could be G, C, G, G. It's as if these 2 are out of order, but they're out of order to form a better D stem. That's the D stem right there. This sequence tests as likely homologous to that sequence there statistically. This sequence here should correspond to CCG, CC, like that. And imagine a C, C, G, C, C. It fits best at these 3 positions here. That's the yellow bit. it corresponds to these 5 right there. So that's one way of solving the problem. That's how I initially approached it. So based on that, this is how tRNA evolved. And this is, so we're going back 4.2 billion years. That's worth mentioning. This is a GCG repeat, 5' acceptor stem, UAGCC repeat, a D loop, CGC repeat, a 3' acceptor stem. That's 31 in all, a D loop mini helix. This is GCG repeat, stem loop stem, CGC repeat, 5' acceptor stem, 3' acceptor stem, stem loop stem. And this is our U-turn loop with a U-turn after a U, followed by presenting 3 nucleotide anticodon. And this sequence here is either identical to that or it's complement, and it's really hard to tell now which it is. If you ligate here and there, which you have to do to replicate mini helix world, you get this longer RNA here. 3 times 31 is 93. This can be part of a larger RNA with complementary replication. And then it can be processed back to mini helices. Okay, so this is intermediate and processing to replicate mini helices, or it can be processed forward to type 2 tRNA and type 1 tRNA in a simple manner. To make type 2 tRNA, you really only have to delete 7 yellow and 2 green, 9 in all. And the same deletion is found in type 1 tRNA. And over here in the V loop region, to make a type 2 tRNA, your V arm is actually a 3' acceptor stem ligated to a 5' acceptor stem initially. Your type 1 V loop is generally 5 nucleotides, and they're the same as those 5 nucleotides. And this deletion takes out 7 green and 2 yellow. And if you're really clever and faster than me, you'll realize quickly that this deletion here and this deletion here are identical on complementary strands. So you have complementary replication, and you can have one processing mechanism to form the deletions. This sequence here becomes the T loop, and it interacts with the D loop, so it's strongly selected after you form tRNA. So if you want, you can look at generation of type 2 and type 1 tRNA as a replication error for mini helix world. So this gives you an idea of what these molecules did in pre-life. I'm going to say that you start with ACCG. That can combine with a GCG repeat, which is what that is, a longer version of that. You can bring multiple molecules together, run wet-dry cycles, and make polyglycine using this molecule. This molecule, this molecule, and this molecule are mini helices. There are 31 to there and 35 to the end if you add ACCA gly, and you can use this molecule to make polyglycine, and this molecule and that molecule. To make tRNA, you ligate this yellow base to the green, yellow to the green, and then you process without showing details, because I'll show them in the sequence, you generate type 2 tRNA and type 1 tRNA. Type 2 tRNA has a single 9 nucleotide deletion right there. The same deletion in type 1 tRNA. This deletion in type 1 tRNA is identical to this one, but on the complementary strand. to process an early version of this molecule into that molecule. You can make a 9-nucleotide deletion that takes out 2 yellow and 7 green and links 5 yellow to 5 cyan, 5 yellow linked to 5 cyan, like that. And there are very few sequence changes that are required to support the novel tRNA fold after many helices, as you evolve from many helices. So just to go over some of this again, you have a GCG repeat, UAGCC, CGC, GCG, stem loop stem, homologous or complementary stem loop stem, and CGC, GCG, CGC. So life is made out of little Lego. And you have this deletion in type 2 tRNA, this deletion, same deletion in type 1. Here you have a deletion in type 1 tRNA that takes out 7 green and 2 yellow. This deletion here is identical to that deletion there, but on the complementary strand. And here you have 7 yellow linked to 7 green. And now you've produced this molecule and this molecule. This is a G right off the rack. That forms the Levitt reverse Watson-Crick pair to a C at the end of the V loop. And here you have a G, which is changed to a C in ancient organisms to form the same interaction at the end of the type 2 V loop. Let's see, here you should have an A, but it's changed to a G. That's our intercalating base. That intercalates between an A, sometimes a G, and another A. Hydrogen bonds to the U before the U turn, the slash, the way I've drawn the sequence. And this is a G off the rack. That forms a GC Watson-Crick pair there. That's the GC pair. That's the intercalating base. That's a G in this tRNA, often an A, and another A, hydrogen bonding to the U before the U turn. Here you have GGC, GG, changed to GGGC, and that can be many different bases in tRNA. And that is to pair back here. So if you have G, 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 C, you make a better D stem. That's the D stem. So that explains this small sequence change here. What am I missing? This sequence here is evolved to form the elbow to the D arm right there. That's the elbow contact there. Okay. You may not believe me that the D loop is based on a UAGCC repeat. So I'm showing a sequence logo to try to reinforce that. So this is just a way of posterizing multiple sequence data, so it's easier for a human. I think that's the purpose of this. Anyway, UAGCC, UAGCC, U, G for A, that's the intercalating base, GCC, UA, almost all the way across the top tick is the UAGCC repeat. This sequence here, GCCU, pairs with GGGC to form the D stem, as I showed you. This forms the Levitt reverse Watson-Crick base pair to a C, as a reverse Watson-Crick pair. This is a G. It should be an A according to the repeat sequence. It's changed to a G. That's our intercalating base. And this G forms a pair to the C. These are elbow contacts here. The main function of the D loop is to form the elbow. Okay, the sequence I just showed you was 17 in length, the D loop. And this is the anticodon loop, stem, and the T loop, stem shown as logos. And if you're used to looking at logos, you'll probably agree that this is complementary to that. That's complementary to that. This loop here is a U-turn loop, and it specifies coding. And this is a U-turn loop that forms the contacts with the D loop at the elbow. You have a U-turn after a U, followed by wobble, Watson-Crick, Watson-Crick. This used to be a wobble position, I'm going to say, and part of suppressing wobbling at this position is modifying this position neighboring. You have a C here and an A there, and they interact and then they stack on the stem. And what that means is that this is a tight loop that resists nucleases in a pre-life world. So it's chemically selected in pre-life. Here you have a homologous U-turn loop with a U-turn after a U. And these bases form contacts that I just described at the elbow. So here you have a wobble position, Watson-Crick, Watson-Crick. So there's something wrong with the wobble position that may be apparent. It's no A in a wobble position at the base of the coat. Can't have the A. And instead you have C, G, and U. G is okay, it'll substitute for A. So G will pair with C, Watson-Crick, and with U, wobble. So G is sufficient, but still degeneracy comes into the code according to wobbling in tRNA. C is a problem. C pairs well with G and poorly with A. And U is a problem. U is a problem because of something called super wobbling. So if you have U in mitochondria in the 34 position, it will pair with A, G, C, and U in mRNA. So if you want to evolve a code with 2 codon sectors, you have to restrain the use of U. It's difficult to expand the specificity of C to interact with A. So U is modified originally by elongator protein 3, which also has transcriptional functions, but it's also an acetyltransferase, or the 5-carbon of U, that then attracts other modifications. So that's to restrain super wobbling. Okay, and U pairs well with A and pretty well with G. So this line here, this was always a Watson-Crick, but this used to be a wobble. And the evidence that this used to be a wobble is at least in part that U is a problem and A is a problem. So if you have A here, you have M1G here. If you have U here, you have T6A here at the base of the code. And as you get some evolution, you get some improvements in that. If you have 2 wobbles, a wobble there and a wobble there, there's only purine pyrimidine resolution at a wobble position. So in evolving the code, you have 2 possibilities and 4, or 2 possibilities and 4. You hang up at 8 amino acids, you can only read 1 wobble position at a time. And then as the system evolves, you go to 2 possibilities, 4 and 4. That's 32. It's not 64 like you may have learned in school. So 17, 17, and the D loop is 17. It's unclear at this point whether the T loop is initially identical to the anticodon loop or whether it's its complement. That's indicated there. They're almost identical sequences. And here's the mini helix. You have a GCG repeat, CGC repeat, 5' acceptor stem, 3' acceptor stem, stem, loop, stem, and these three are scrambled from coding. And this system is set up to synthesize polyglycine. And you could probably do that in your lab using published procedures. Okay, here's the anticodon loop. This is stem, loop, stem. It's a tight loop. This is an AC pair. It's a modified C. This is a reverse Hoogsteen interaction that stacks on the stem. So this is a tight loop that resists nuclease degradation. Then you have a U followed by a U-turn. That's sort of easy to remember. The U-turn is really important. It has to be a U-turn loop here. And almost always this is a U. At the base of the code, it's always a U. Then you have Bobble, Watson-Crick, Watson-Crick. But as I explained, modification of this base here is part of the reading of that base. So this is a tRNA phenylalanine. This is an A, and that's a modified G. This is a eukaryotic tRNA. And so this is WY-butysine. It's really a complicated G modification to read A. To read U, you have a complicated A modification at this position, T6A at the base of the code. That's one of the ways that wobbling was suppressed at this position here. So this I'll describe kind of quickly because I'll go through this a couple of ways, but maybe the thing to do is to introduce the problem first. So this is a tRNA leucine. And so it's got 7 yellow plus 7 greens from an ancient organism. And LURS reads a UAG sequence on the end loop of the V arm. Okay. And with CRRS, you have a different trajectory of this arm from the tRNA body. And you have contact here to the yellow part, the green part, and that part. So the problem is that leucine and serine are in 6 codon boxes. And so what that means is that there are 5 tRNA LU for the aminoacyl tRNA synthetase to recognize. And so it does not do it by viewing the anticodon loop. Instead, it looks at the V arm, which is consistent in the set of tRNA loops. And for tRNA-seer, there are 4 of these, but again, too much diversity in the anticodon loop to use that as the signal to accurately aminoacylate. Instead, it uses the arm and it uses a different trajectory of the arm to attach serine. So in archaea, leucine and serine are the only 2 amino acids that utilize a type 2 V arm. So you're limited by the number of type 2 V arm tRNA sets you can use by the trajectories of the loop that have evolved. So type 2 T-arms, I'm going to say, are a 3' acceptor stem ligated to a 5' acceptor stem. tRNA-seer in this organism has a 1 base insert. And this is a 3' acceptor stem, a CGC repeat, and that's a 5' acceptor stem, GCG repeat. And this is after 4 billion years in this organism. Type 1 tRNA should align to the first 5 positions, like that, derived from this sequence here. Okay, so we should do this little snippet there and that little snippet there, and then we're pretty much done. So 5' acceptor stem, GCG repeat, CGC repeat, 3' acceptor stem. These 5 should correspond to those 5 right there. The sequence should be GGC, GG. Instead, it's GGC, GG with these 2 bases flipped. And they're flipped to form a better D stem. That's the D stem right there. And this little bit here should have the sequence CCG, CC, and that would be CCG, CC. It's best at this position and that position, maybe not too badly at this position. But it tests as likely homologous to this sequence there. Okay, back to this question of tRNA loos and tRNA sears in archaea. So in archaea, leucine and serine are 6 codon boxes. Arginine is a 6 codon box, but it's a different story. So the rule is 2F by Lou and 1F by sear, which is sort of like an old Longfellow poem about Paul Revere's ride, which I'll get to in a minute. But that gives the trajectory of the arm. Okay, so 2F by Lou, 1F by sear. There are 4 of these and 5 of these. They all have about the same arm, same secondary structure. Type 1 aligns like that. This UAG interacts with LU-RS1A. And you see the problem. There are 5 of these, so 5 different anticodons here, and really consistent sequence of the V arm for recognition by LU-RS1A. And SEER-RS works similarly. Well, it's a different enzyme, but it works and about qualitatively in the same way. Okay, so here it is. 1F by lambda and 2F by C, Paul Revere's ride, and 2F by Lou and 1F by sear and archaea. Bacteria, it's like a mess. We don't want to talk about that right now. Okay. So here's LU-RS from an ancient organism, and it's interacting with UAG right at the C terminus, this beta hairpin here. Contacts very strongly to the G and the A, a little bit less with the U. The U is the least conserved of the 3 bases. If you look at more sequences, no contact at all to the anticodon loop. I think it's an interesting problem how contacts here and at the elbow, that's our GC pair at the elbow, how that gets communicated here to accurately amino isolate the tRNA. So it's a problem for molecular dynamics simulation, I think, among whatever other methods can be devised. That's LU-RS. And SEER-RS is a very different enzyme. It's an alpha-2 dimer. It's type class 2 instead of class 1. And it uses this N-terminal helix hairpin to lie across the yellow part of the V-arm, the green part of the V-arm, and interact with the elbow like that. And it's reading the distinct trajectory of the arm from tRNA loop. And this is a bacterial structure because I couldn't find an archaeal one. This is good, and it's got the wrong trajectory of the arm as a result. So it'd be nice to do more with this. There seems to be negative cooperativity binding tRNA sear. There's no tRNA sear bound here. I had to steal this from another structure. Oh, and I forgot our little logo at the end, ACCA. Okay, so this is a little Lego piece. I'm going to say that ACCA with glycine at the 3'O is the primitive adapter molecule. In tRNA, this base becomes the discriminator base. It's usually A, the oldest um, uh, sequence, and then reluctantly G and more reluctantly U, only for threonine and cysteine, and C for only for histidine. But there's a little bit of a problem if you have three C's in a row, and that is in the peptidyl site of the peptidyl transferase center, you have two G's base paired here and here. And so if they base paired here and here, that would be a problem, and you'd be misaligned for forming peptide bonds. So the way evolution has gotten around that is to add GTP as a post-transcriptional modification of tRNA his, and it soaks up this C and forces it to use these two C's for alignment of in the P site. Pretty clearly, this modification is as old as the genetic code. There are a lot of these tRNA modifications that are as old as the genetic code. So what does all this mean? So without having really emphasized this in my talk, there's evidence in tRNA sequence for polymer world and mini helix world, and you sort of go in this progression. And these three worlds can coexist initially, but at the time or about the time that tRNA learns to code, tRNA goes back and mostly eats mini helix world and polymer world, because that's how evolution works. And but embedded in tRNA is still sequences that indicate this world and that world. So what's this all about? So I'm going to say that it's all about synthesis of polyglycine. I don't know whether it's clean or dirty polyglycine. That's an interesting question, but maybe for another day. Anyway, this is an artist's view of polyglycine, and I'm going to say that it has two roles. One is to help you on this progression from lipids to protocells to cells, and the other purpose is to accumulate macromolecules as they would be accumulated within a cell in a modern archaeal or bacterial cell. So you get polyglycine mini helix aggregates. You get small molecules such as cofactors like S-adenosylmethionine that can protect single-stranded RNA in a pre-life world. You have lipids going to protocells, going to cells. In tRNA, you have GCG, CGC, and UAGCC repeats. That's a complement of that, so you probably have the complement of that. And here you have stem loop stem that becomes the anticodon loop, and possibly, well, probably its complement, which may have become the T loop. And you have ACCA gly as the primitive adapter, and you have ribozymes, and they can be stabilized by S-adenosylmethionine. So this is beginning to look like a complicated world with a strong chemical selection for polyglycine. This is probably too many words, but I'll try. So the pre-life world was a competition for the clumping of advantageous macromolecular aggregates, as on the previous slide. So polyglycine, RNA, cofactors, ribozymes, metabolism. Life evolved around tRNA and the tRNA anticodon. You start with ACCA gly, and if you mix this with a GCG repeat that's pretty long, you can get multiple ACCA glys in proximity, can run wet-dry cycles and run the dehydration that's required to form polyglycine. And then you start ligating this to all sorts of different RNAs until you get to tRNA. And the reason you do that is it's a better way to form polyglycine. So you're driven by polyglycine synthesis. I guess tRNA could leap forward in a frozen accident and start Darwinian selection, but I don't think it happened that way. I think tRNA starts out forming polyglycine and then goes from that to lysine, alanine, aspartic acid, and valine, the four simplest. These are all in the same row, row 4 of the genetic code. And then you add leucine, serine, glutamate, and arginine. Leucine, serine, and arginine form 6 codon boxes in the eventual code. And these two did it using type 2 V-arms, as I described. In concept, arginine could have used the same strategy by picking another trajectory of the arm, but it didn't do that. Instead, what arginine does is it unwinds the anticodon loop to expose more bases. But what I want to tell you is that if you try to use this strategy for all 3, that won't work. Okay, so there are too many constraints on the sequences that are used to recognize tRNA arginine. And if you get these amino acids, you can use tRNA linked chemistry to convert serine to cysteine, glutamate to glutamine, aminotransferase, aspartic acid to asparagine. So you're locked up at 8 amino acids. I sort of showed you how that happens, but you can add 3 to that, and you can start to make first proteins. From here, you go to 20 amino acids plus stops and a genetic code and what I'll call LUCA, the last universal common cellular ancestor. And now you have Darwinian selection, and you can go back and eat a lot of this preliminary stuff. The code starts in tRNA with the anticodon, gets cast into mRNA as the codon, and then stored in DNA. And all this stuff can evolve along with the genetic code, can co-evolve. So I'm going to say life evolved from a complex RNA amino acid and RNA peptide world. And this is really learning without foresight. So tRNA learns to code, but it learns stupidly, like a computer learns or like evolution learns. It learns along this pathway by trial and error. And polyglycine has 2 roles, RNA peptide cofactor aggregator, and also support for co-evolving protocells. So a strong chemical selection for polyglycine. And these are just a whole bunch of examples of things that are in that pre-life world and co-evolved with the code. And here's a longer list and a longer list, and that's all published if you want to look at it in more detail, or I can discuss it in questions. Recent publication where there's an attempt to order the amino acids into the code, and I've attempted the same thing, but what I would say is that this is kind of incomprehensible, and so is this. But if you break it into code columns, then it makes much better sense. And I don't want to go through this in detail. I just want to give you a sense for how this works. I don't know how I'm doing on time, actually. But if you break it into code columns, then you can begin to understand that this is actually a very orderly assembly. So this is the archaeal code, which I'm going to say is the simplest code. And you have in columns related amino acids, related charging enzymes, aminoacyl tRNA synthetases, related amino acids, related charging enzymes. I'm going to say this one used to be a 2A, and then before LUCA changed to a 2D to discriminate from some of these other amino acids. Here you get this lovely stripe pattern, orange, green, orange, green, orange, green, like that. Aspartic acid was in first, and then glutamate, which is a related amino acid, is here. This uses a 1V charging enzyme, 1V and 1V. And this is a 2B, a 2B, and a 2A. This one deviated from that one, and then that one was derived from that one. This one was derived from that one. That's an interesting story, but not one I'll tell today, I guess. And here you have ARG as a 1A and cis as a 1A related aminoacyl tRNA synthetases. You have aromatics going across this row. That's a disfavored row because it's 36A. You have to suppress wobbling at 36A. You do that with the M1G modification in the next position, as I described. This is 36U here in row 3. That uses T6A at the base of the code to help suppress wobbling at this position. So you need this modification and that modification. You need to modify U if you're going to use U, and you do that using the enzyme elongation protein 3, ELP3, plus other modifications on top of that to suppress super wobbling so you can have 2 codon sectors in the original code. A isn't utilized in Archaea, and C is lightly modified for elongator met, unmodified for initiator met, heavily modified for ILU, and that results in the killing of this tRNA right here. And there's more to be said about this, but I can't remember what. So 1, 2, 3, 4, 5. There's a 6 modification here. Lots of tRNA modifications just to build the code. I went over that before. The counter arguments to what I've told you are kind of dumb, but some of them have made their way into print. They might be such as a 7 nucleotide GCG repeat is not a 7 nucleotide GCG repeat. That's a 7 nucleotide GCG repeat. A complementary 7 nucleotide GCG repeat is not a 7 nucleotide CGC repeat, CGC repeat. A 17 nucleotide UAGCC repeat is not a UAGCC repeat. That's a spit there. 2 homologous 17 nucleotide stem loop stems are not homologous. They're obviously homologous just by inspection. And so let's see, with Bob from physiology, tried to figure out type 1 tRNA evolution, and we figured it out, and it was pretty much proven by Daewoo Pak doing statistics on the model. I know you can't prove anything with statistics, but we came close at least. And Bruce out of the blue emailed me and told me how this evolved based on what we'd published about this. And he was right, and we co-published with him. And my former graduate student, Lei, now a professor at the University of New England, Biddeford, Maine, and he continues to interact with me on this project. I hope Chris and Yensu are making lots of money now. They did a lot of work on this. And now we're getting some help from this gentleman at Northeastern. So those are the topics. And that's what I'd like somebody to do next. Probably not me because my lab has been closed. So I'll stop there and take questions. Thank you. David. Very big step into a ribosome. I know that Carl Rose had things to say about that. I don't remember what they were. What are your thoughts about that? Okay, so how do you evolve something that's the kind of molecular horror that is the ribosome? And what my thought is the ribosome is actually a very simple initial construct with a whole bunch of add-ons. So I think the job of the ribosome is sort of to provide a peptidyl transferase center that's a dehydration chamber to support the addition of many different amino acids to the growing chain. But then ribosomes have become very complicated since then. Lots of things to maintain proper stepping through the structure, and tRNAs flow through a tRNA-shaped channel. So there are all these kinds of bells and whistles that are added after. But I think the initial ribosome is simply a construct to synthesize polyglycine, and then it learns from there to synthesize more complicated products. Right, so are you asking, so I would say that ribosomal RNA was created by a very similar process to what I showed for tRNA. And I would guess that tRNA was one of the constituents in the original ribosomal RNA. Finding those homologies has been a little bit of a challenge. People claim to have done it, but I'm not sure that they've done it in the best possible way. So what I would like to see is somebody looking at ancient archaea and ancient bacteria and finding some of the same tRNA homologies in both of them and seeing this extent to which they relate. That would constitute proof for me. But I think that probably ribosomes for ribosomal RNA was made out of a junkyard that includes tRNA fragments. Let's see. Probably no useful thoughts. Right. So when you get out of phase on the bacterial ribosome, there's a termination mechanism, and I'm trying to remember some of the details, but it involves a tRNA alanine derived molecule. I forget what it's called. And it's involved in the release. So I thought that was interesting there, but there are a whole bunch of things like this where tRNAs get, you know, brought in for other purposes and do other jobs. GADV. Yeah. Well, people have looked for that a whole lot, and if they've had great success at it, it's unknown to me. So I've read some of those papers, and it seemed to me like those kinds of records might be overwritten based on what amino acids subsequently are selected for best function. But I think GADV is like a tangled polymer, probably involves mono amino acid polymers and copolymers of various sorts. How those get sorted out, I don't know. But I think that they're an improvement on polyglycine as an emulsifier of cell components. That's how they get selected. Oh, how could you? Yeah, so basically I gave you a scheme where ACCA gly gets attached to various RNAs. Those RNAs have to evolve to tRNA. So you could start with tRNA polymers, GCG repeats, CGC repeats, UAGCC repeats, stem loop stems, and you could start to figure out how to assemble a tRNA molecule, how to utilize ribozymes to assemble that molecule in vitro. So that's what I'm advocating for. I haven't done that experiment myself. You'd have to synthesize them. Right. So I think what I showed you gives you a lot of ideas for how to go back and cook up the original GCG repeats, CGC repeats, UAGCC repeats. Then you have to slice them and dice them and build 31 nucleotide mini helices that then you have to combine to make tRNA. So I don't know where I would start experimentally. I might try just with the initial creation of tRNA using a ribozyme and ligating 3 mini helices together. So if you could get that processing to work, that would be probably a pretty good paper, I think. Okay, so not that I know of, but it's very easy to make amino acyl tRNA synthetase ribozymes, very easy to make ligase ribozymes and endonuclease ribozymes. People have made those in vitro very easily. Yeah, right. And I showed you that you can make more, you don't have to make them as small molecules. You can make them as large molecules because the ligation and replication mechanism generates large molecules, large like ribosomal RNA. Thanks, Ed.